dear distinguished speaker, Professor Yonath, dear honorable guests, colleagues, students, ladies and gentlemen, good morning, and welcome to the University of Macau. It is our great honor to have your presence for the conferment ceremony of Dr. honoris causa on Professor Yonath, Nobel laureate in chemistry, in recognition of her remarkable contributions to biomedical sciences. The University of Macau confers this honorary title on individuals who have accomplished a significantly high caliber of achievements in science that bring great contributions to mankind, to the community, and to the university. As a world-renowned scientist and expert in ribosome research, Professor Yonath's research works are highly influential to the medical sector and have significantly promoted the development of new medications for improvement of overall living standards of humankind. And for her pioneering discovery in the structure and function of ribosomes, Professor Yonath was awarded the Nobel Prize in Chemistry in 2009, and more principal awards for her research contributions in this field. In addition, Professor Yonath had been a member of the International Advisory Committee of the University of Macau and is currently a member of the Advisory Board of the Faculty of Health Science of the University of Macau. As a young university undergoing expansion in research and teaching, all of us in the University of Macau are very thankful to the renowned scientists and experts like Professor Yonath, who spared the time to share the research experiences with our faculty members and to give advice on the development of the faculty. Indeed, in recent years, through the efforts of the university community, growing regional and international cooperations, and inviting world-renowned scholars to visit and collaborate or deliver lectures and talks in our university, we have made several breakthroughs in various fields. For example, we have 5 fields with papers that have made the required number of citations in the past 10 years to become world's top 1% in essential science index. The engineering, pharmacology and toxicology, clinical medicine, and computer science, social sciences. Also, our progress is being recognized in world university rankings such as QS and THE in recent years. Hence, we will continue to work hard to create conditions favorable for the steady growth and development of the university. And we'll devote more effort on attract top scholars, establishing research facilities, conduct international leading interdisciplinary research, etc. We hope to see more distinguished experts and researchers visiting Macau and the University of Macau in the near future. Today, we are very honored to have Professor Yonath here with us. She is going to deliver a lecture titled Next Generation Species-Specific Eco-Friendly Antibiotics and Thoughts About Origin of Life. It is one of the many examples how her magnificent research work has shed light on medical development for better life qualities. I'm sure that our audience were highly inspired by Professor Yonath's lecture for better understanding on how origin of life is linked to antibiotics. Ladies and gentlemen, please give a big hand to welcome the 2009 Nobel laureate and our honorary degree recipient of Doctor of Science, honoris causa, Professor Yonath.
It is a very, very big honor for me to give this talk and also a pleasure to come back to Macau, although my way here was not so simple because of the airlines. So what I want to discuss today is essentially what we are doing now, so many years after we already saw the structures. But in order to start, I want to acquaint you with the ribosome. So DNA is the cellular instructions for how to make proteins. This is a typical DNA piece, double helical, that all the information about how to make proteins is here in the bases that are combining the two helices. Here the information is given, but it cannot be read by any biological molecule because it's hidden by the structure. Therefore, the first step is to translate this to RNA, and RNA has the same number of letters, the same language as DNA, only 4 letters in the genetic language of everything that is done in the world. 4 letters only. So all biology. And every 3 of them, or every 3 here, are coding for one of the components that make the proteins, which are shown here, which are called amino acids. And this is done by the ribosome. So this is what was interesting for me. All ribosomes are universal. The function, it means the reading of the code and the making of the proteins is almost the same in all living cells. Doesn't matter where they come from, universal. So ribosomes exist in every living cell, bacteria, fish, flower, trees, human beings, animals, everything that lives is having ribosomes in it, in every cell. And they are reading the instructions and making the proteins according to the instructions. As I said, 4 letters, every 3 of them are coding for 1 amino acid. There is a huge number of ribosomes that function in every living cell. In mammalians, like in our liver, can be 4 to 5 million ribosomes in one cell. Even in bacteria, they can reach up to 100,000 in one cell. In vivo, it means in the cell, they function continuously and they can make 40 bonds. The bonds that connect the amino acids in the proteins are called peptide bonds. They can make 40 of them in 1 second, and they hardly make mistakes. So I was the fastest student in my class. I finished the lab exercises the first, not because I was so good, but because I was very poor and I had many jobs and I had to finish fast to do my jobs to exist. So I was the first. It took me 6 hours to make 1 bond. This was the fastest in the lab. Now chemistry developed and it takes 2 hours to make 1 bond, but not 40 in 1 second, as ribosomes do. Now that we know how ribosomes work, we like to describe them as factories that have 2 floors. In the top floor, the instructions are being read. It means the RNA that brings the instruction. And in the lower floor, the amino acids, if it is a factory, then they are being brought by trucks. So the trucks come here with the amino acids. I'll talk about the trucks in a minute. And when there is here a 3 nucleotides, the signal for this amino acid, it will come in and connect to the newly born protein. This happens in the factory in the lower floor. Then the truck will be empty and will go out. The protein will be longer by 1 amino acid, and the message will progress in 3 letters for the next amino acid. This is all. It takes a little bit of energy, not much, and this is all. Very simple. And it's being done all the time in every living cell, also us. And it took me and my group 20 years to find it out. And at that time, 2 more groups. So I just want to say a few words about the tracks. The tracks are also RNA molecules, which are called tRNA. They always have the same outer shape, like L, with here the 3 nucleotides that have to combine with the message here. And on the lower part, the amino acid that they carry that is cognate to these 3 nucleotides. And this part, and this part are the specific ones. This is the common part, which is almost the same in every living cell and for every amino acid. This is the important one for decoding. And this is the amino acid that has to be connected. And always in this part, which is also called the 3' end, always is the same sequence, CCA, fully conserved. So when we got the structures, we decided that we want to make a short clip, which I want to show you now. Can I get the lights lower? Dim the lights, please. And this clip is available in YouTube. There were hundreds of thousands of people that saw it, that hitted it. And we put it in 2013. It means already 14 years ago. And so far, I got no complaint. It means probably it's correct. That this is the way we think that the ribosomes in bacteria work. This is what we knew at that time, and also we know today, of course. So we did it together with 2 students in the Art Academy in Jerusalem, and we focused on 1 ribosome, although they usually work in polymers, in many ribosomes. So what you see here is the messenger reaching the small subunit. It comes to the small subunit and binds to it. It adds a path, and it is all moderated by factors, this blue thing on the right side, which is called initiation factor 3. Now that it is bound and everything is fine, the first track, the first tRNA can come and find its position. It's clear that in order to make bonds, we need at least 2 tRNAs, so you will see the first one coming, and then we continue. The first is also brought by a factor. It's called initiation factor 2. The cell has factors to start the ribosome function, because the cell doesn't want the ribosome to work alone. It wants to be in control of it. Ribosomes are made of 2 subunits. What you see here is the small subunit, where the decoding happens. And once the small subunit is happy, the large subunit can come and join. The large subunit is where the bond is being made. So this is the small subunit with the first tRNA bound. And now comes the large subunit and makes bridges and binds to the first one, so there is a working ribosome. And we will look at it from the solvent side. The top one is the large subunit. And you can see how more and more tRNAs are being brought by elongation factors. The ribosome helps in, out, 40 times a second. And meanwhile, there is a decoding that is being done on the small one, and the proteins are being made on the large one. We took away the large one. We left only a tunnel. You see in the lower part of the picture. You can see now better. The newly born protein is going out through a tunnel that protects it. So the process continues. This protein, this blue thing down there, can fold, and when everything is finished, there is a code that says finished, and then instead of tracks come this yellow thing from the left side. This is a factor that terminates it. It's called recycling factor. There are also release factors, and the whole process finishes when the two subunits separate the protein. The blue thing down there comes out, and the machinery is ready for the new protein. That's all, ladies and gentlemen. So simple. Only 20 years, minus 2 weeks. So what you saw in the movie and in the factory, top floor, the small subunit, lower floor, the large one. Decoding happens in the small subunit, made in the large one. And all of this is made by tRNA, the tracks. So you can see it here again. Top one, the small subunit. Large one is the bottom one. The amino acids are being decoded here. The tRNAs are this yellow, red, and green. They are reading the amino acid here and producing in the large subunit the new bond here. And the position where the new bond is being made is called PTC, peptidyl transferase center. So you can see easily that there are 3 positions for tRNA in the ribosome. The first one that was occupied is in the P site, in the PTC, in the peptidyl transferase center. The second one that came is the A site, amino-acylated tRNA. And the third site is for the exiting tRNA. So the names that are common, and I will relate to them, are A for amino-acylated and P for peptidyl. And now I show again the positions where things are happening, reading and producing. So until now you didn't see details, but actually we know the position of each and every atom in the ribosome. In the beginning of my work, nobody thought about such numbers, but it's actually 250,000 atoms. The ribosome is made mainly of ribosomal RNA, which is the gray thing here, and many ribosomal proteins. What you see rotating is the small subunit that in all bacteria has more or less the same molecular weight and sediments as 30S. A molecular weight is 0.85 megadaltons, one RNA chain of 1,000 nucleotides and up to 21 proteins, 20 to 21. So this is the small subunit. To remind you, the decoding happens here, in this narrow place here. And the large subunit is also shown with the same color code. To remind you, it makes the bond, a peptide bond. It elongates the protein. It means it is a polymerase. It's also made of RNA and many proteins. Molecular weight 1.5 million daltons, 3,000 nucleotides, and up to 34 different proteins. Each protein here is shown in a different color. So because of the fundamental role played by ribosomes, and now you know how fundamental it is, it's the genetic code translator, many antibiotics target them. Over 40% of the clinically useful antibiotics target protein biosynthesis, mainly by paralyzing the ribosomes. You may know or may not know that the natural antibiotics, which are actually the main useful antibiotics, are the weapons that bacteria from one type is using to interfere with cell life of different species, different microorganisms. It means bacteria against bacteria. This is the war from the microorganisms' life, microorganisms' creatures. And they are very, very effective. So first question to answer is, how do the tiny antibiotics paralyze the giant ribosome? The ribosome, as you saw earlier, is 2.5 million Dalton, and the antibiotics less than 1,000. How do they do this? Very clever. Very, very clever. Each of them is targeting, is binding to another functional site. So if you see here the small subunit and the large subunit, these circles, each of them is representing a family of antibiotics. So let me just tell you what is a family of antibiotics. The bacteria produced an antibiotic, whatever, and companies, when antibiotics came into use in the middle of last century, jumped on it and make them better, improve them, and so on and so on. But they stayed with the skeleton from the antibiotics. So these are different families with their names here and here. And look, all of them are binding to important functional places, like the decoding, the bond, the tunnel, and the hinge. The hinge are these motions that you saw. Very clever. And in order to show you how clever, we made another little clip, also available in YouTube, which is based on the first one. And it shows a few antibiotics. I will show you 4, 2 that go to the small subunit, like this one, Edein. Look how clever. Very small, but very clever. Huh? That's it. No message, there will not be protein. Or the second one that is called tetracycline, that occupies the position of what we call the A site tRNA, this little green thing. Just doesn't let the tRNA bind. That's all. One is not enough for a bond, so stop. And now you can see two antibiotics that go to the large subunit. The first one is called Erythromycin, is actually the first from ribosomal antibiotics that went into use. Sits in the tunnel and doesn't let protein go. Only very short protein, 5, 7 amino acids, and that's it. And the last one is, from the chemistry point of view, the cleverest, goes into the bond, into the position of the bond. Oops. Isn't that wonderful? So there are some questions. All ribosomal antibiotics bind to ribosomal functional sites. You could see 4 of them that we just saw. There are 30, but I showed 4 representatives. But these are highly conserved. All ribosome perform translation in the same way. And we don't want to kill the patient. It's mandatory for clinical use to distinguish between the pathogen, the bacteria, and the patient. And if the sites are similar or almost the same, how does it happen? So sometimes there are subtle differences, and I want to show you one. And the whole usage of antibiotics is based on this. So what you see here is looking into the large subunit, into the tunnel with erythromycin in it from top. So you see that it blocks most of the tunnel, not the entire tunnel, but most of it. If you look at it from the side, if we cut the ribosome this way, the large subunit this way, this is the large subunit, this is the tunnel, protein would be made here and would continue if erythromycin does not stop it here. I think it's clear. So this part will not be. How does this happen, the stoppage, the blockage? So we look at the tunnel in this position. If this is the tunnel direction, perpendicular to it. And this is what we see, a pocket, the walls of the tunnel. And they are made of RNA, only RNA, the gray, as the color code before. And what you see here inside are many, many antibiotics from the family of erythromycin, which is called macrolides. So these are all macrolides, all are built on the skeleton of erythromycin. All bind into the tunnel. This would be the tunnel if it's empty. And you see they block the tunnel either excellently or only very good, but they block the tunnel. And all interact with one of the tunnel components, this one, which is A, adenine in position 2058 according to E. coli. The type of binding is shown here. Very high affinity between erythromycin and this nucleotide A, 2058. Very, very high. I think a very, very good chemist or bioinformatic person would not be so successful in the affinity here. Very, very high affinity, which is actually love between the two of them. Now, ladies and gentlemen, please focus on this point here. And you will see the difference between bacteria and us. Bacteria and higher organisms, that's all. But when there is a G here, it's too close to the erythromycin and it doesn't let binding. That's all between bacteria and us. Another problem with antibiotics is resistance, and I'm sure that you all know that resistance is a big, big problem of modern medicine in the whole world. I'm not telling you a new thing. And what can we do about this? So first, let me tell you, for those of you that don't know, that resistance is a basic process. The bacteria know to do it themselves, or microorganisms know to do it themselves, regardless if they already saw medicine, Western medicine or not. It means the fact that doctors prescribe prescribe antibiotics helps resistance, but did not start resistance. Actually, all these papers describe attempts to understand how resistance was made, and they found that it's done by the bacteria themselves. So one of the, I will focus on one of the delegations that are described that went into the jungles of Venezuela just a few years ago. You see, just recently, 2015. They found that the people that are living in this jungle, the tribe, never saw any Western medicine or Western food, yet in the bacteria of their own life, the bacteria that they carry that are called microbiome, they had antibiotic resistance genes. This is their own way of the bacteria of this tribe to fight against other bacteria. So it's, or to resist against other bacteria. So these findings show that resistance to antibiotics is a basic process for the survival of many microorganisms, regardless of their exposure. And it suggests that microbes have long evolved the capability to fight toxins, including antibiotics, and that preventing drug resistance may be harder than scientists thought. They are right. So before I go on, I want just to show you one of the ways that bacteria developed specific molecular pathways to cause resistance. And it's based on this process that I just showed you a minute ago of the selectivity. With A, there is binding. With G, there is no binding. It's not a big deal for the bacteria to rotate themselves, to mutate themselves and make themselves with G. And now they are resistant. So this is the resistant by the bacteria themselves, one of the ways, not all the ways. So changing from A to G by the bacteria. So the increasing appearance of multi-drug resistant strains, together with the minimal, actually negligible number of new antibiotic drugs that are presently undergoing development in companies, is a real big global problem. And it seems that soon we will come back to the pre-antibiotic era, when people died from pneumonia or wounds at the age of 50 or even earlier, like Mozart, for instance. Even the World Health Organization said we are coming to post-antibiotic era. The antibiotic will not be useful. And even the bank, the World Bank estimated that up to 3.8, almost 4%, be will be gone, be lost by 2050 because of resistance to antibiotics. Yet, as I said earlier, very few new antibiotics are in development, hardly any, by companies. And most large companies stopped attempts because they are afraid of resistance and because they think that there is a very low cost of them. It means there is a huge mismatch between the social value, it means our life, and the expenses. So first, let me tell you what I think about, or let me tell you about what I think about combating resistance fully, resistance to antibiotic fully. I think it's unlikely because bacteria want to live and because they are cleverer than us. They are very clever. So to make new antibiotics means to be more clever than bacteria. And this I will explain in a minute. But first, I want to remind you that most pharma companies stopped making antibiotics. And I want to show it to you in a graph. This is the growth of resistance, normal resistance for these free pathogens, free bacteria that cause diseases. And this is the number of new antibiotics. Less than 4 antibiotics in last 20 years in all companies. So we decided to do something about it in our lab. Maybe we can contribute something to this fight of the world against bacteria. So until recently, most of the available crystal structures were of pathogen models. It means of bacteria that are not pathogens, they are not causing diseases, but behave like them. It means they showed the common modes of antibiotic action, but they didn't explain specificity, what makes a bacteria being a pathogen or not. So we crystallized the whole ribosome from several pathogens, including Staph aureus, which is a very multi-drug resistant strain, very causing many diseases. And we compared these ribosomes to the normal ribosomes. And we got some lessons. So I will not tell you about all lessons in detail, but just some of them I will highlight. All bacterial ribosomes are similar, but not exact. This was lesson 1. General lesson, sorry. Lesson 1, it is possible to increase antibiotic effectivity. There is for example, there is a company in Vienna that increased the effectivity of non-antibiotics by a factor of 25, by changing a little bit the chemistry. Lesson number 2, in principle, it's possible to increase antibiotic selectivity. We found specific positions in the ribosome that can be used to increase the discrimination between different species, pathogens and not pathogens. We didn't do anything about it, but we suggested it to the companies. So far they didn't answer. Lesson number 3 I'm skipping, and I'm going directly to lesson number 4 because this is the most exciting lesson that I have, beyond my expectations. And this is novel antibiotics binding sites. So first of all, let me show you the comparison between normal ribosome or large ribosomal subunit in gray and a pathogen. You see, there are very few little places here that are larger in the pathogen. There are also some other places that are shorter in the pathogen. I don't talk about them now. But those are less than 10% of the whole, even less than 5% actually, of the whole ribosome structure. So statistically, and the statistic people told me it will not be of any use. But when we saw the structures, we think there is a use. We think that these 10% have specific structural motifs. Let me focus on this one, the lower one here. You can see here the model, the pathogen model in gray, and the pathogen, exactly the same here, exactly the same here, and here, and here, and even here. But here there is a little extension, very small, only 9 nucleotides extra of the 4,500 in the whole large subunit. But it is a feature, it's a structural feature that may be a potential binding site for new antibiotics. So we actually found many of them, no, not so many, but some of them. In the large you just saw, and you can see it rotating, and also in the small, which I don't show. So using a model system, a model bacteria, we identified 25 new potential unique sites. We blocked them chemically, and all of them, and 16 of them inhibit protein biosynthesis. And these sites are really, really very good because they are not involved in the primary ribosomal activity, namely decoding and peptide bond formation. Hence, no pathogen contains any genes for their modification. So they are new. And they are so new that here we beat the wisdom of the bacteria. This means that since there is so far no vital role that was assigned to this bacteria to these ribosomes by the bacteria, for now, this can be exploited for design of advanced degradable antibiotics, hence environmental friendly. So actually I didn't talk at all about environment and degradable and selectivity. So let me talk about it for a minute. First of all, what is the difference between selectivity and similarity? So I'm sure that you all know what is the microbiome. These are the trillions of bacteria, the flora that are existing in the flora of our body in semi-exposed organs like guts, ears, lungs, and skin. They are extremely useful because they are involved in the health of us, and they are all susceptible to antibiotics. It means when we take antibiotics against a lung problem or a wound or something simple, if the antibiotics can advance into these regions, like the guts and lungs and so on, the antibiotics destroy the microbiome. Also, I want to talk about selectivity. So pathogens against the microbiome, so far there is very little distinction, and I will come back to it in a minute, and versus eukaryotes. So versus eukaryotes, our new sites are very suitable because the growth from bacteria to eukaryotes is shown here in the addition of the red. It's almost all on the surface, and our sites are on the surface of the pathogens, so they will not be found in eukaryotes. So the selectivity here is really very good. So I'll come back to the microbiome in a minute, because first I want to tell my opinion about ecological problems. So almost all ribosomal antibiotics have inside them pieces that are resistant to digestion. They are not being digested by us. And when they go out of us, like everything else that we don't digest, into the sewage, in many cases, the filtration and the purification systems catch them because they are so small. So they come back to us through the grass that the cows are eating, which means their milk, or through other plants or trees that grow them. So we get them back. We get these internal pieces back to us, although they don't, we don't need them, We don't need antibiotics when we get it just from the water, and they increase the resistance. So our new ideas, new positions, can give us ideas for new antibiotics, novel antibiotics, that are not in the potential sites, and therefore we can do them as any chemistry we want. We can make them in chemistry that we can optimize against antibiotics action and against degradability. We can design them as we want, for degradability actually, not against. We can compose them from nucleic acids or polypeptides or oligopeptides, the way we design them. And this is what we are doing. Design antibiotics that we can control their chemistry and their degradability. So in summary, our new insights obtained from these high-resolution structures of ribosomes from a genuine pathogen, actually now we have several, give us unique tools for suggesting novel sites, I just showed you, for suggesting how to improve the clinical performance. This is what the company that we collaborated with did. For better distinction, that we have the chemistry of it, but so far no company wanted to take it. And for the design of pathogen-specific antibiotics. Pathogen-specific antibiotics. What does it mean? It's like personalized medicine. Each pathogen will have its own antibiotics. So it will be possible once the pathogen is identified to give just one antibiotic or a few antibiotics that are specific for this pathogen and not take the wide range antibiotics that are used now for many. So we are aware of the problem, the economical problems with the companies that don't want to make antibiotics and surely are against pathogen-specific antibiotics. Nevertheless, we expect that the pharma companies will reconsider their suggestions, especially when they will realize that many people die in young age before they use their very expensive compounds, very expensive therapeutic compounds that they produce. So efficient use of pathogen-specific antibiotics means fast and smooth identification of the type of the relevant pathogen. And it's not so simple for the doctors to do it in hospitals, but nevertheless, steps towards this aim are already in progress by some medical professionals and by some companies. So we hope that this will be possible. Actually, it's already possible to do it within a few hours for some of the antibiotics. So the use of the production of specific pathogen-specific antibiotics also requires many structures. Each structure of each pathogen. How will we do this? By X-ray crystallography. Took it, as I told you, 20 years for the first one, and later on 2 to 4 years for each. This is much too long. Luckily, single particle three-dimensional cryo-electron microscopy became recently useful. Actually, it's the Nobel Prize of last year. So with this, within a few months, we can get very, very high quality, high resolution structures. So we tried it first on the ribosome from Leishmania, which is a disease that is not very serious usually, but when it is becoming serious, it is fatal. And 12 million reports were reported last year of it. It's eukaryotic. It's transferred by a fly, the sand fly, and it's common in the south side of the world, including Israel and Macau and so on, all here, all the blue one. And within a few months, although there is very little chemistry known, we could determine the structure of this ribosome, all the proteins, all the RNA, I'll show you the level of accuracy. These are all the proteins. And if you see here, this is how accurate we could see each and every atom. Also in the RNA, we could see, we could draw it this way or that way, but we could see each nucleotide. We could see magnesium ions. We could see modifications like that or that. Actually, we could identify 99 new modifications in the RNA of this ribosome, which gives us a possibility to target these specific ones and not others in the patient and so on. So in short, our structure-based species-specific design aims at minimizing the widespread resistance, minimizing microbiome destruction, and minimizing environmental hazards. So a small bonus for you, which I already told you, is some thoughts about origin of life. Do you want to hear it? So just to remind you, these are the active sites in the small subunit, the decoding in the large subunit, peptide bond formation. And when we look at them in the ribosome, we see that the decoding and peptide bond formation are residing in RNA environment, protein-free, no proteins here, no proteins here. It means all the catalytic events of the ribosome is done by RNA. And this means that ribosomes is an RNA machine. And the question is why? Why does the cell, until now, use RNA to make proteins? In principle, proteins could make themselves. So we think that this may be a very sophisticated regulatory machinery. Two kingdoms. RNA produce proteins. It is also in good agreement with the ribosome structure. Mainly all ribosomes, except for mitochondria, have RNA to proteins 2:1, in ratio 2:1. Even in mitochondria, when part of the ribosome, of the RNA in the ribosome has been replaced by proteins, still the active sites are the same. And also it may mean that the beginning of life was RNA, that it dominated the prebiotic world. Why do I say this? This is what I want to explain in the next few minutes. RNA, before I go there, RNA enzymes today are known to be lazy, lousy, not working well, but nature could make efficient biological machine to make proteins even from this lousy thing. So just to remind you, proteins are made here within the ribosome. The ribosome here is in gray. The A site tRNA is in green, in blue, and the P site in green. This is the position of peptide bond formation here. And I will keep the blue for A and the green for P throughout the next few minutes. So what was found first is the ribosome does not do any catalytic event, just provides the frame for making the proteins here. And all what it provides is the stereochemistry of the A and P tRNAs to produce peptide bonds. And what you can see here is a superposition of many structures of substrates of the ribosome that were determined either in our lab or in other labs, competitor labs. All are the same. Doesn't matter if the substrates were long for this side or long for that side or long here or large here. They all sit in the active site. They sit the same. And this is in good agreement with what I said earlier, that the ribosome provides a frame for making peptide bonds, And this is in good agreement with other studies, biochemical studies made on all these places, which I will not talk about each of them, but all show that ribosome just either places the substrate or increase the function of the substrates themselves to activate the reaction. And this frame, as you can see here, is semi-symmetric. It looks like this is symmetrical to this frame. So we looked at the whole sequence and the whole structure of the large ribosomal subunit. In sequence, there is no symmetry. There is no part that is symmetrical. But in structure, we can see symmetrical a behavior here, semi-symmetrical, like there is a twofold symmetrical axis here, and each part here of the main chain, not the sequence, can be rotated around here and come to here. This is like a twofold symmetry, and if we look at it in more detail, the top part is hosting the P-site tRNA, and the lower part is the A-site tRNA, and what I didn't tell you until now is this is where peptide bond is being made. The level of symmetry is if one puts the green on the blue, this is the amount of symmetry. You can see it this way or that way, but anyway, it's symmetrical. So the peptide bond formation site within the contemporary ribosome looks like that. The A site that hosts the A, you can see it here with the A, tRNA, the lower part, of course, of CA tRNA, and the green hosting the P site here. And they are exactly the same everywhere. If we look at it from top, it's exactly the same. Of ribosomes from eubacteria, archaea, eukaryotes, mitochondria, these are the names of them in the data bank. Look, exactly the same. And this is this part of. And if we look at the level of conservation of all the 180 nucleotides, we see that most of them are 100% or close to 100% conserved. Some are 50%, but very high level of conservation all over the place, much larger than the whole ribosome. This is just this piece, the symmetrical region, which we think that its high conservation hints that it indicates that it's beyond environmental conditions. It did not develop, it didn't evolve like the entire ribosome at all, from bacteria to us. So we've proposed that this is the Proto-Ribosome, where the first peptide bonds were made even in the prebiotic time. Which the Proto-Ribosome originally was a prebiotic bonding entity that we call the Proto-Ribosome that is still functioning in every ribosome, in us and in every ribosome today. And just I write it again in an ancient way, an RNA apparatus with catalytic capability functions within the contemporary ribosome in all of us. Just to show you where it is in the ribosome, this is the large subunit. It is here. These are the two tRNAs that combine it to the small subunit. And this is its combination to the two hands. You remember in the movie? So it's actually sitting in the heart of everything. These were the pieces that moved all the time to get in tRNA, to get out tRNA. These are those that move. And, of course, it makes the peptide bond. So, question is, we know that peptide bond is being made here, but when? And I want just to show you how small this piece is. You see here the large subunit, and this is the piece of it. 3 to 4%. Here you can see its position with space fill if you want. tRNA doesn't really matter, and this is what we saw all the time until now. And that's, of course, the structure of it. And this is where it is in the whole ribosome. Large subunit, small subunit, this is the part connected to the small subunit through the tRNA. This is its shape from top and its shape from the side. So this is actually what we call, what we call the Proto-Ribosome. So based on the suggested existence of RNA-dominated world, and on the finding that RNA can replicate and elongate itself and can have catalytic capabilities, we propose that not always this is the beginning of ribosome, that we propose that Proto-Ribosome is the entity around which life has evolved, the beginning of life. So in order to show it, we try to construct it in the lab. And I just go through several slides that show what are the properties of RNA. So about RNA-dominated world, it's here. Neither of them is ours. About synthesis of ribonucleotides by RNA itself, all this works. Neither of them is ours, I just cite. About RNA enzymatic activities that may be very important or are very important to our work are here. And how RNA can activate and make substrates of RNA-bound amino acid, it's here. So I hope that I convince you that all what I told you is now documented in the literature, and therefore I can come back to our proposition that Proto-Ribosome is the entity around which life has evolved. What are the possible pathways for this? Again, I want to remind you, RNA can replicate and elongate itself. So, as I promised you, lab constructions that we try to make it. One of our hypotheses how this region was created is that it was made of two parts. This catalytic Proto-Ribosome was created of two parts that are more or less shown even in the sequence today. This part and this part. So we produce such parts in the lab. Pieces of RNA that according to the RNA-dominated world that suggestion could have existed then. They can come together and make pockets that can make. So we can make these types according to the structures today or according to structures that we can predict that will fall like this, just any structure. So as I said, fold like this, or take it, try to dimerize them. We found that not every pair A-site regions, like here, did not dimerize. A and P didn't dimerize. P alone dimerized. Look, we can see here, P alone dimerized. So only P, which is showing that there is a non-uniform tendency to dimerize. And this indicates that survival of the fittest and natural selection seem to play a major role even before life. It means what we see here is spread Darwinian Darwinism, not of animals or organelles or organisms, but of molecules in a prebiotic world. So now we have that we have some dimers, we try to make peptide bonds in the lab. This took 9 years, and last February we got it. So what you see here is peptide bond formation, normal, just to remind you, 2 amino acids are making a bond. In ribosome research, usually amino acid is used, 1 amino acid is used, and 1 mimic of amino acid called that is called the, oh God, I forgot the name, I'll come back to it in a minute. But it has an amino acid part in it. It's an inhibitor. And making a bond here says ribosomes are active. And we found it in the lab. We made a bond here. Right here, we got now a peptide bond in the lab. Took 9 years. But if you want to know a little bit about it, you can see it here. The RNA chain, the Proto-Ribosome is 67 times 2 bases from the P1, which you saw earlier. The amino acid, the D amino acid was identified by MALDI. We did several positive and negative controls. We don't know yet how long it takes. And we had to stop this experiment for a little while. It was done in February because the postdoc that did it went to give birth to her first baby. So now she's coming back to the lab and she will continue, and I will be able to tell you more about such bonds. Before I finish, I want to thank the Weizmann Institute that let me work years when I was called the dreamer. The president made the decision, Michael Seller. He's a biologist, so people said it was of biology, but it was not changed by Chaim Harari, that is a physicist that replaced him. They were encouraged by the Scientific Advisory Committee, 2 Nobel Prize winners, Kendrew that determined the structure of the first protein, hemoglobin, myoglobin, and Anfinsen that showed that proteins work fold according to their sequence. And Alex Rich, who tried to crystallize ribosomes for years and years and failed, he was still supportive. So I thank him twice as much. It all started in a very intensive collaboration with Max Planck in Berlin, with their director, Dr. Wittmann, who wanted really to understand how ribosomes work, and he was very essential in starting a little research group for us in the synchrotron where we measured in Hamburg. He died 10 years before the structures came, and he was replaced first by Franceschi and then by Fucini. So I had 2 groups, one that worked in Hamburg and one that worked in Weizmann Institute, and I want to thank all the members in both groups for their determination and devotion. The group in Hamburg stopped working in 2004, and the group in Israel, including some people that have already left, is shown here. So the group is actually run by Dr. Yonath Bashan, who is the major scientist, and it includes Ilana, who found the symmetry in the active site, and the rest of the people that work a lot on antibiotics, and Tanaya, who was able to create the first peptide bond in the lab. I also want to draw your attention to Tamar. Tamar came 16 years ago for 10 weeks, and she's still around. And the day that I took the picture, she had birthday, and she baked a cake. So first of all, female scientists know to bake. Second, you want to see the cake? Here is her cake. We chose that in my group, ribosomes are considered sweet. I also want to thank my family, mother and sister and so on, and daughter, who is an MD, and granddaughter, who was at that time a kid. Then I got a prize in Paris, UNESCO prize. She made a speech for me. She said, my name is Noah, Ada's granddaughter. As you know, she is a very busy scientist, but she always finds time for me. And she gave me a prize for it. You want to see her prize? This is not her prize, this is the Nobel medal. But for me, her prize is at least as important. The grandma of the year is Ada Yonath. And when I asked her which year, every prize has a year. She said, which year? You have to reprove yourself every year. And if you fail, I'll take it off the wall. And I want to show you still on the wall, 10 years. She gave it to me even before, more than 10, 12 years. Even before I got the Nobel Prize. It's still there. And this is the only prize that I can lose. All the other prizes are mine forever. She can take this off. Ribosomes became popular in Israel. Here is me. And she is wife, look, they are dancing. She's the ribosome. Protein comes out of her. And also young kids. And an artist, very good artist and writer, Michel Kishka, made this from me. it says head full of ribosomes. But if you look at it, small subunit, large subunit, symmetry axis, and positions where he thinks best antibiotics should bound. Thank you.
And first, I'd like to thank again to Professor Ada Yonath. This is a great honor to all of us. The ribosome structure we learned for our undergraduate studies. As a professor, I teach molecular cell biology. I talk about the small subunits, the large subunits, how to make protein. But that time, I didn't know the story of Ada Yonath. I didn't know that took her 20 years to finish this work. And also at this age, she carried on her research.
I am 21 years old.
Yes. Always.
You are still 21 years old.
Yes. Always 29 in Hong Kong. We always say that.
Because you have good ribosome.
Yes, and also, and her work probably will move the industry about making new antibiotics. And so now I have the first question and to Professor Ada Yonath. That there are many new sites that is existing in pathogenic bacterial cells, not in the regular ones. And can you say how stable those sites, those mutations?
I think the mutations are very stable. The proof is that these mutants are, the resistance mutants that have them are more stable than the father molecule, the father bacteria. And some of them, if you let me one speculation, some of them, when we look at them, are almost identical to extensions that happen with evolution between ribosomes from bacteria and from yeast. So we think that maybe pathogenicity, which is not in ribosome, but the fight against pathogenicity is the missing step between prokaryotes and eukaryotes. It's even more than just sites. But I think that these sites are, in my opinion, are very stable.
Thank you.
Maybe there were many, many other sites, but they were washed out by the bacteria.
I was surprised that the 16 out of the 25 sites you tested inhibited, because they're not in any of the regions that are what you would have called the major regions.
Don't call. This is the major regions.
Oh, so wait.
No, they're not in the major.
That's right. So
yeah.
so how do you think they're actually inhibiting?
Okay,
you might have guessed that that wouldn't have worked.
So we looked at them. We looked where they are. It's not, I just showed you, but I didn't analyze one by one. It's a lot of studies. They are all in positions that can help the ribosome be created. It means between the large and the small subunit, so they can eliminate the production of the ribosome, or in positions that interact with the factors, the elongation factors, the initiation factors. So even in bacteria, they are important for the cellular production of proteins. But even if we do it in vitro, in the test tube, we always have these factors. And when they don't let them bind, okay, one peptide bond maybe can be made accidentally, but not a protein. So they disturb protein production, but not by disturbing the major, or what we call the major role of the sites with the major role, or the activity, or the vital sites. There are several words for this.
So I think that must also mean that those sites, even though they're different, must have evolved to help stabilize the ribosome in those new pathogenic bacteria. Otherwise, they wouldn't be affected.
Well, yeah, they contribute to being effective. And out of the 25, we know about 15 what they do.
In terms of trying to prevent resistance with your new approach to antibiotics. Given your wide knowledge of the structure, can you predict which sites would be less likely to lead to resistance, or do you think that this is mainly an empirical issue to see whether resistance develops?
Prediction is not my part in science, and there are many predictors that are trying to do it, and I wish that they come out with results. From the bacteria point of view, they don't really predict. They try. They have a generation is about 4 to 6 times a day, so they can change themselves, mutate themselves with almost no limit. And those that exist, that survive. So what they can, evolution that they can undergo in one day for 4 generations, for us is hundreds of years. If a generation is 25 years, 4 generation is 100, 6 is 150. Means they just try. I don't think they think.
So since the antibiotic resistance become a crisis, a global crisis, and you also showed that about 10 million people will die by 2050.
Yeah.
So my question is that, do you think eventually we can overcome the crisis? And another question is that the pharmaceutical companies are reluctant to develop new antibiotics. And so do you think academia or government should take more leading role on the new antibiotic development?
The price of antibiotics when they sell it to people is not very high. It's not like $20 million new therapeutic for cancer that they can charge $10,000 per one pill. It's relatively cheap. Antibiotics are relatively cheap, and they are used only for infections. Means they are not everyday prescription like the elderly people things against they take as cocktails against blood pressure and insulin and cholesterol and all types of elderly people maintenance, which are very good income. So for the companies to decide to put 10, 15 years in developing a new antibiotic and at the end sell it with low price and for a short time it's not profitable. And they explained it to me. So for having new investors, it's not a good idea to talk about antibiotics. But when I told them that in my opinion people will start dying again at the age of 40, 50, and will not reach the age of their customers, their money-making customers, then they started to think a little different. And maybe there is a hope in this direction.
I was just wondering if the RNA part is the key, it seems the key of the whole ribosome structure, then why does the life take so much energy to make the whole protein part? Is it just to protect the delicate machine, the RNA structure inside? It looks like the structure of a grapefruit to me. The fruit inside and the thick skin and everything outside seems to be just protecting the things inside. But if that's the case, then why the ribosome structure is like the grapefruit but not the orange or the apple, which has a very skinny skin?
I think that the ribosome is much more complicated than just giving a pill, which you said just protection. It's much more than protection. The structure is so complicated in order to be efficient, fast, correct. There are so many proteins that keep the structure together inside. They have long tails. I will show you what I mean. And these are keeping this machinery to be oh God, I passed it. To be efficient and correct and to reject mistakes, not just a pill. It's much more than a pill. And why grapefruit? You see, I have in my garden, I have oranges. Now maybe I'll think about changing them by grapefruits. Maybe they're more strong. But otherwise, I'm happy with what I have. And the ribosome seems to exist billions of years. So what I mean by proteins, these are all the large subunit proteins in this ribosome. And you see, all of them have globular parts. These are on the outside, but they also have long tails or internal loops. And they penetrate into the structure and hold it together and help it fold and assemble. They have much more than just a pill. They are much more. And probably nature, if it's so happy in all organisms to have the ribosome structure this way, at least the internal part, probably it tried many other things. And we don't know because they don't have a lab protocols. It means not all what they tried we know, we just know the result.
I want to add one more point about ribosome RNA, because we know ribosome has RNA and many proteins. Those proteins actually greatly stabilize the ribosomes. And for example, the messenger RNA are very easy to be degraded, but the ribosomes are very stable. And I think those proteins also plays a role to stabilize the ribosomes and made it into a machine to make protein.
Thank you, fully agree. This is a good answer to you.
Thank you.
I think we have the last question. I think we have the time is very limited. All right.
Okay.
Oh, you have.
Yes.
Then
Professor
you
Yonath.
and then also have another one. Okay. Have two questions.
Yes. First of all, welcome to Macau, and I know you had a very long journey, but you got here finally. So thank you very much for giving us such an enlightening talk. My question is also about the origin of life. Why do you think that nature decided to use RNA to make protein and use protein to make RNA and other nucleic acids, at least polymerizing them? Why can't RNA make itself?
It can make itself, but it takes much longer time. It can make pieces, and the gluing of the pieces is more difficult, but it can make. RNA can elongate itself. For what we think in the beginning of ribosome, RNA was the genetic code for the RNA.
Yes.
Yeah. Why the nature did this or that? I think nature tried many things. And this is what was best functioning. Because both RNA and proteins can do the job, and RNA just selected the best combination. Nature selected the best combination.
Nowadays, the RNA world still exists, considering billion years evolution. They may have trace evidence still exists in some species of medium species on Earth.
Yeah, the Proto-Ribosome still exists for billion years. This part is still existing.
Yeah, but the direct evidence is lacking right now. Many study has been done in the lab, in the test tube, and since Craig made this proposal 50 years ago, my question still is argument is there any RNA world which physically exists in the world?
The piece of the ribosome physically exists in the world. The rest I cannot answer you. As you said yourself, there is no direct evidence. I am only 21 years old, so I don't know what happened 50 million years ago.
Okay.